So at the end of the day, yesterday, I was talking about handling and use of the microscope. Okay, and we said that obviously the carrying technique is one hand on the arm, one hand on the base. Okay, the microscopes all have these dust covers. They used to actually be like real dust covers, but they all tore. So now they're Ziploc bags. Okay, but they cover the microscope. Always make sure that one goes back on yours before it goes back in the cupboard. Okay, because dust is like a major enemy of microscopes. Okay, we talked about how the stage can be moved with the little knobs here. Okay, we talked about that there are 3 different objective lenses. Now, which lens should you start with? Right. You should start with the scanning power lens in place. That's the short one with the red band on it. The reason for that is this. Okay? As you move up in magnification, okay, the area you can see decreases because you're blowing it up bigger. All right. So if I ask you this question, let's say you're watching a Flames game. So you're on one side of the stands, okay? And you know your friend is somewhere on the other side, okay? You have a pair of binoculars with you for some reason, but you do, okay? What would be the best way to locate your friend? To scan with the binoculars, okay? And oh, sorry, I forgot to mention your friend is wearing a bright green shirt, okay? Everybody else is wearing red like they always do. Your friend's wearing a bright green shirt for some reason. Okay, would it be better to put on the binoculars and try and scan through every single kind of row or person in the crowd, or to just kind of look in general until you find something green and then put on the binoculars? Which would be better? Scan, then put on the binoculars. If I put on the binoculars, how much can I see? Much less, right? I'm seeing a much smaller area, much more greatly magnified, but I can only see maybe 20 people at a time, okay? Whereas if I'm just kind of looking for this bright green shirt in general, I can see basically the whole other side of the stadium, okay? And I have a much better chance of going, okay, that looks green. I'm going to put my binoculars up and look at that green thing I just saw, okay? That's what we want to do with a microscope. We want to start out with the scanning power lens. Okay? The scanning power lens sees a very large field of view. Okay? The area that we can see through the microscope we call the field of view, or FOV. Okay? On the scanning power lens, it's very big because the scanning power lens doesn't magnify very much. So it's much easier to find the thing you're looking for. You're not going to see any detail on that thing at this level, but you'll be able to find it easily. Then you'll move up to the next level of magnification. Okay, so you'll go from 30 times magnification to 100 times magnification. So now I only see that much area. Okay, I see it in much more detail, but because I've blown it up, okay, I can't see anything that's out here anymore. Okay, does that sort of make sense? And when I go to the high power lens, I'm looking at that spot. Greatly magnified, but that's the only spot I can see. Okay, so we want to start on the scanning power and then move up in magnification as we go. The other thing we need to do as we're using the microscope is make sure that each time we move to a new magnification, we put the specimen where? In the center. Okay, now our microscopes, thankfully, have a pointer in them. Okay, so anytime you look through the microscope, you're going to see this line. It's actually a very fine wire that's inside the ocular lens. Okay, it's supposed to be there. Okay? I once had a group who kept bringing me microscopes that were broken. I'm like, what is wrong with this? There's a big black thing in there. Oh, okay. I'm looking at it, I can't see. They bring me another. There's a big black thing in this one. They brought me like 3 microscopes. I'm like, the pointer? Was that supposed to be there? Yes. Okay. So the big black thing that points towards the center is supposed to be there, not broken. Okay. All right. So each time I find something and get it focused, I want to use these little knobs here to move the stage so that I can get that on the center, okay, or in the center of the field of view. Because when I move up in magnification, if I don't, and the specimen I'm looking at is out here, I won't see it because I'm only magnifying this area now. Okay, does that make sense to everybody? Okay, so I've got to always make sure that I center it before I move up in magnification. All right. Okay, and obviously we talked yesterday also about using the lens paper to clean the lenses. When you are done with the microscope, you should return the microscope with the scanning power lens in viewing position, because that's the one the next person will need first. Okay, it also is the one people assume is in position, and we want them to, we kind of want them to do that. If they assume that that one's in position and we leave the high power lens in position, they'll crank the high power lens with the coarse knob through the slide, okay? If they're really not paying attention. But all right, just make sure that the scanning one is back in. So we got scanning, low, and high. Those are the three lenses we have. Now, some things you'll be expected to do, okay, with the specimen that you view under the microscope. Okay, you will be required to estimate its size. Okay, this is actually very easy. All right, all you have to know is what is the field of view for each lens. So you're going to need to write that down. Okay, so the field of view for the scanning power lens, which magnifies, sorry, I said 30 times, but it's 40 times. Okay, the field of view is 4,000 micrometers across. Okay, this letter here is not an M. Okay, micro, okay, is mu. It's the Greek letter M. Okay, we can't use M because M already stands for millimeters, so we have to use mu, micro. Okay, when you're typing this out, you can actually insert that symbol if you want, but I will also accept a U. Okay, because it typically looks like a U. Okay, if you draw it right, it kind of looks like a U there as well. Okay, so mu m, okay, or micrometers, it's 4,000 micrometers across. That's 4 millimeters. Okay, so when you're looking at the scanning power lens, okay, you are seeing an area that is the distance between my two fingers. Okay, that's how big across the field of view is for the scanning power lens. Okay, it's tiny. All right, like you barely get like a a letter from a newspaper in a regular article to fit, okay, in there. All right, so it magnifies reasonably well, but not a lot compared to the others. All right, so if I'm looking at an object on the scanning power lens and it looks like this, about how big is that? Hmm? Yeah, I'd say somewhere between 1,000 and maybe 1,200 micrometers. It's an estimate. We're not calculating it. Okay, estimating the size of the object works just like that. Okay, this is 4,000. That's about a quarter of it. Okay, it's about 1,000. That's good. Okay, that's all you have to do to estimate the size of an object under a microscope. Okay, it is, after all, just that, an estimate. Okay, now for the low power lens, this distance is 1,600 micrometers. Okay, because we're magnifying it more, right? So if we see a smaller area, now it's only 1,600 micrometers. Okay, and for our high power lens, obviously we see an even smaller area that's only 400 micrometers across, right? Because, well, it's 100 times more powerful than the scanning power lens. Sorry, not 100, 10 times more powerful. All right, everybody okay with that? Good. So as long as you've got those numbers, you'll be able to do your size estimates. And the reason size estimates are important is they're part of our lab diagrams that we make, okay, after we look at specimens under the microscope. Okay, so we got those there, and we talked about that already. All right, I want you guys to quickly answer these questions. I'll give you about 5 or 8 minutes or so to work on those, okay? Get some complete sentence answers for those questions, and then we'll talk about them here in a couple of minutes. All right, so what are the 3 points of the cell theory? Harrison? All organisms are made up of cells, okay? What else? Cells carry out the basic functions of the organism, probably the most important point of the 3, okay? And the third point is? Right, cells are not created, they come from the division of existing cells. So those are the 3 points of the cell theory. All right, be a really easy multiple choice question. Which of the following is not part of the cell theory, as there are 3 options there and 4 in total, okay? Question number 2, who are the 4 people considered to be the most important in the development of the cell theory? What did they do? Okay, first person would be the guy who discovered cells, and that would be who? Hook, okay? The doctor, not the captain, okay? Okay. And then after that came the guy with the ping pong paddle shaped microscope, Leeuwenhoek. Okay. And then the 2 guys who actually proposed the theory, from Sweden, Schleiden and Schwann. Okay, those 2 guys. Okay, so they're the ones who proposed the original cell theory. And then third question, on what basis can we deduce that plants and animals may have had a common ancestor? Their cells have similar structures, not just structures, but also processes. I mean, yes, structures is important, but also a lot of the cellular processes, biochemical reactions and things like that are the same, okay, between both types of cells. And what? Yeah, like cellular respiration. Yeah, the way that both plants and animals metabolize sugar. All right, we both require oxygen for that. We both produce carbon dioxide as a byproduct and water. Okay, all of that kind of stuff. The big differences have to do with the mode of nutrition. Plants can make their own food with photosynthesis, and animals have to consume food. All right, so we'll leave it at that for that one.